Welcome to the first video in the bacterial genetics series. The first part of this topic is going to focus on DNA replication, which is how a cell or an organism copies its DNA, because all new cells that are made must have an intact copy of the genetic material, which for all living things is DNA. So this video is just going to be a quick refresher on DNA. So let's first remind ourselves that DNA is a nucleic acid. Okay, that's what the NA here means in the title, nucleic acid. Same thing with RNA. Okay, so it's telling you what type of macromolecule it is in the name. Now, all nucleic acids are made up of nucleotides. Okay, and so in this picture, here's a nucleotide. So we're going to have our nitrogenous base, and then we're coming up on the 5-carbon sugar, and then finally the phosphate group. Okay, so those are the 3 individual components that make up a nucleotide. Now let's circle the next nucleotide. So I'm going to start over here by this second phosphate group, coming around by the sugar, and then another nitrogenous base. In this case, it's adenine. So there are 2 different nucleotides, and in a strand of nucleic acid, there's going to be thousands of them, okay? So again, all nucleotides are going to have those 3 individual pieces. And I want you to notice how those 2 nucleotides are connected. They're connected right here through a bond between the top sugar and the next phosphate group. Okay, and so you can see the connection between nucleotides 2 and 3 starting here. Okay, and so again, it connects the sugar of one nucleotide. Oops, sorry, I meant to make that a pin. It's going to connect the sugar of that second nucleotide, and that would connect the phosphate group of the next sugar. So here's the sugar, it's a little 5-carbon sugar. There's an oxygen there. And then here would be the nitrogenous base. So for all the nucleotides that make up DNA, and they all have the same sugar, deoxyribose, they have the identical phosphate groups. So what sets the 4 different nucleotides apart from one another is the nitrogenous base that's attached. And so the 4 bases are guanine, cytosine, adenine, and thymine. And so when we talk about the sequence of DNA, it's the order that the nucleotides are arranged in. And so we'll just refer to them as A, T, C, and G, just for simplicity. Okay. So notice that the sugar in the top nucleotide, right, is connected to the phosphate group in the second nucleotide. Then the sugar of that second nucleotide is connected to the phosphate of the third nucleotide, and this would continue on. And so because of this is how each individual strand is held together, we say that DNA has a sugar phosphate backbone. All DNA has a sugar phosphate backbone. In fact, so does RNA, but we'll get to RNA later. The next thing that I want you to look at, let me erase these pictures real quick, is how the 2 strands are connected to one another. Okay, so if you look at the nucleotide that I was just looking at right here, okay, you'll notice there's a nucleotides on both strands. So the one on the left-hand strand has guanine as the base. The strand on the right has cytosine as the base. And look at how those 2 bases interact through hydrogen bonds. Y'all, that is super important, okay? Because during DNA replication, we're going to have to separate these 2 strands of DNA. And so we need them to be held or connected to one another by the weakest bond possible, which is a hydrogen bond. Okay, so just to reiterate, the sugar phosphate backbone that holds all the nucleotides together that form a single strand is a covalent bond, sugar phosphate backbone, very strong. The type of bond that connects the 2 individual strands together is a hydrogen bond, because we need those to be able to break apart easily. Okay, so the next thing that I want you to notice here is the directionality. Okay, each of these strands have a unique direction, okay? And so if you're looking at this left-hand strand, the bottom is called the 3' end, and the top is the 5' end. But then look at the second strand on the right. Notice it runs in the opposite direction, right? It has its 3' end at the top and 5' end at the bottom. So what does that mean? Well, it means that the two strands are anti-parallel. Okay. And so they're like two one-way roads, right? One of them is running left to right and the other is running right to left. So you might be saying, Chelsea, why on earth do we call these 5' and 3'? Well, that has to do with the picture of the sugar that you see on the bottom right. So the sugar is the middle component of the nucleotide, okay? Notice that the phosphate group is attached to the left and it actually attaches, let me erase this word anti-parallel real quick. All right, so on this 5-carbon sugar, on this 5' carbon is where the sugar is going to be attached. And on the number 1 carbon is where the base is going to attach. And for some reason, it's not letting me write on there. But on that number 1 carbon at the 3 o'clock position is where the nitrogenous base attaches. And so if you look again at that top picture, look specifically at the top of the left strand, the 5' end. Do you see how the topmost molecule is going to be this phosphate group, right? That oxygen is sticking up at the top. At the bottom here is the sugar, okay? Specifically, there's a hydroxyl group, an OH group. Sorry, y'all, OH group. And that's the bottommost molecule of this particular strand. And so in order to kind of let, a reader or a student, whoever, know, the 5' end always has the free phosphate group at the top. That's the topmost molecule. And it's got a free hydroxyl group at the bottom at the 3' end. But now look at the right-hand side, okay? At the top of the right-hand strand is that hydroxyl group coming off of that sugar. And we know that the hydroxyl group is from down here at the 3' position. So it's called the 3' end. And then this free phosphate group, which is always attached to the number 5 carbon, is at the end of that molecule. So wherever the free wherever the free phosphate group is, that's the 5' end. Wherever the free sugar is, that's the 3' end. Okay, so that's what we mean by directionality. So the two strands of a DNA molecule are always anti-parallel. And there's one more feature about these strands of DNA that you need to remember, and that is that they are complementary, okay? And so this refers to base pairing. Anytime we're talking about DNA, the base adenine will always pair with the base thymine, which is what you see in the top right picture, and guanine will always pair with cytosine, okay? Always. And so why does this matter? What is this, how is this helpful to y'all? Well, what's going to happen on quizzes, exams, and in the process of DNA replication, by knowing the directionality of a strand of DNA and the nucleotide sequence, so write this down. If I told you that one strand of DNA runs 5' to 3' and has the sequence TACCGT, because you know that the other strand is anti-parallel and complementary, you should be able to tell me the directionality and sequence of this second strand of DNA. So take a second while I sit here and talk slowly and write out the directionality and sequence of the second strand of DNA, because you will have to do this later. All right, so we know that the two strands are anti-parallel. So because the given strand runs 5' to 3', that means the other strand will have to run opposite. So that's going to be 3' to 5'. And now we need to do base pairing. So thymine in the top strand is always going to pair with adenine. We know that adenine always pairs with thymine. So again, looking at the third nucleotide in the top strand is cytosine, and we know that cytosine is always going to pair with guanine. So again, cytosine will pair with guanine. Guanine will pair with cytosine. And then our thymine will pair with adenine. Okay, so your second strand is 3' to 5', ATG-GCA. Okay, so this is how you can apply your understanding of the general features of DNA. Okay, the last thing I want to really remind you all of is that DNA is going to be double-stranded in living things, but in prokaryotes, it is circular, and in eukaryotes, DNA is linear, okay? And I guess actually one more thing. DNA is made up of thousands and thousands of nucleotides, okay? But DNA is divided into segments called genes. And a single gene codes for a single protein. That means the DNA sequence of a specific gene provides the directions needed for the cell to make a single protein. So there will be hundreds and hundreds and hundreds of genes on a single piece of DNA, at least for eukaryotes. Okay, right? Prokaryotes have a smaller number of genes than eukaryotes do because we're a bit more complicated than a single-celled bacteria. But the same gist still applies. Okay, the bacterial chromosome is broken into pieces, not like physically broken, but divided into regions called genes. And those genes will each code for their own protein. All right, y'all, that's the end of this video. Let me know if you have any questions.